Good afternoon, everyone, and welcome to today's class. As usual, we're gonna wait for 5 minutes so that people can join in. Okay, and we're gonna discuss about the cell adhesion molecule, adhesions. So we're gonna continue our discussion wherever we left in the last class. Okay, yeah. I guess we can start now. So, yeah, so as usual, what we were discussing in the last class was about uh, cell adhesion markers, or how cell interact with each other. These are the different types of CAM molecules, cell adhesion molecules, and then now we'll see what is the role of exactly cell adhesion. Okay, so assembly of strong cell adhesions require changes in the actin cytoskeleton. In their mature form, adherence junctions are enormous protein complexes containing hundreds to thousands of cadherin molecules, packed into dense, regular arrays that are linked on the extracellular side by lateral interactions between cadherin domains. The assembly of these junctions over a large surface area is not simply a matter of adding more cadherins to an initial attachment site, but also requires changes in the underlying actin cytoskeleton. Most important, strong adhesion requires a decrease in cortical tension at the site of the adhesion. Cortical tension in a cell is much like the surface tension of a water droplet. In water, binding between water molecules at the air-water interface pulls the surface inwards, resulting in a spherical shape that resists disruption by other surfaces that do not interact with the water molecules. Similarly, an unattached cell in suspension assumes a spherical shape because of cortical tension, which results from the contractile activity generated by bundles of actin and non-muscle myosin II at the cell cortex. Just beneath the plasma membrane, this cortical tension is so strong that when two cells initially interact through binding of cadherins to on their surfaces, cortical tension prevents the spreading of the adhesion surface and the cells interact at a single point. So, do you understand what it means to say? It basically is saying that cortical tension in a cell is just like a surface tension of a water droplet, right? So, all of you have seen a water droplet. So, when there are different water droplets, water molecules at the air-water interface pulls the surface inwards. That is why how the water droplets are formed. Then that spherical shape, it has attained so much of tension that now it will negate any changes which you want it to make on the surface, right? And which is not interacting with the water molecules. Similarly, an unattached cell in suspension assumes a spherical shape because of cortical tension, which results from the contractile activity generated by bundles of actin and non-muscle myosin II at the cell cortex. Assembly of a large adhesion surface, therefore, depends on local reduction of cortical tension, which is achieved by inhibition of cortical actin myosin fiber formation. These changes depend in parts on two small GTpases called RAC and Rho. Signals generated by these GTpases govern local actin filament behavior. Rho generally promotes the formation of actin myosin stress fibers at the cell cortex, while RAC inhibits the formation of these fibers and instead promote branched actin network. So we're going to see in the coming slide that there's a diagram how basically actin remodulation helps in the adhesion junction formation. Okay, so for actin remodeling, both RAC and Rho GTpases are used. So what RAC does, RAC basically helps in the polymerization of the actin myosin fibers in the linear direction, while Rho, it inhibits the polymerization of actin myosin fibers in the linear direction and hence promotes the branching of these fibers. Okay, when two epithelial precursor cells first interact at a small cluster of cadherin linkages, the cadherins generate intracellular signal that promote local activation of RAC and inhibition of Rho. This results in disassembly of actin myosin fibers and loss of local tension. which then allows further cadherin recruitment to the spread the adhesion to a larger surface area. So we'll see the figure first. So this is what it means. So imagine these are the surface molecules which are supposed to be pulled inside. This is just an example how a sphere is made, and all the cells are also spherical in nature if they are in a so if, for example, if they are in a liquid environment, right, because of the surface tension. Now what exactly is a cortical tension? So cortical tension is used, it's a term which is used for the actin myosin arrangement inside the cell. So whenever there is a lot of tension in the cell, tension means that there is a lot of pressure inside the cell, and which is balanced by the pressure from the outside of the cell, so that cell can retain in its position and shape. So as I mentioned, for actin and myosin, both RAC and Rho GTPases are used, right? So RAC basically allows the actin filament, for example, these are actin myosin filaments, right? So RAC allows the linearization of the actin filament, and Rho does not allow this, and hence it helps in the branching of these actin or say intracellular microfilaments. So this is the basic procedure of it. So we'll go through the literature one more time and then we'll go back to the diagram. So RAC activation has the added benefit of stimulating local actin protrusions from the cell, which also contributes to expansion of the junction, right? The implications of this mechanism are clear. The development of strong adhesions depends not only on the cell adhesion molecules themselves, but also on the associated regulatory systems that governs actin behavior, which means our RAC, Rho, myosin, these intracellular filaments. Okay. Eventually, after large number of cadherin molecules are aligned at a nascent adherence junction, RAC is inhibited and Rho is activated. So you remember about the GTPase, how G proteins, how we have studied their cycle, right? So G proteins, there is a GDP bound inactive state, GTP bound active state. Similarly, RAC and Rho belongs to such family of proteins, where GDP bound state will be inactive state and GTP bound state will be the active state. So now if we are saying that RAC is inhibited, meaning that RAC is now in GDP bound form, okay, and when we are saying Rho is activated, which means that there is a guanosine exchange factor GEF, which now has activated the inactivated Rho factor with the GTP bound Rho protein, okay, which promotes the assembly of linear contractile actin filament bundles that link the adherence junctions to the actin cytoskeleton. These new linkages pull the junction inward, generating tension that stimulate further actin recruitment and expansion of the junction, as we can see here in the figure. So first one is just the example which we try to understand that how a sphere is formed, basically depending upon the tension or the surface tension of the water molecule, surface tension generated by the water molecules. In the second, we understood that how the very control of the RAC and Rho GTP and GDP bound states are used to assure that the cell has its very tight conformation and it's very well structured. Now when we talk about the formation of adherence junctions, we basically, for example here, these are the two epithelial cell precursors first interact, small cadherin clusters. These are very small cadherin molecules, and you know, if we talk about classical cadherin molecules, they will be homo type of connection, okay, homophilic. So when two epithelial cell precursors first interact, small cadherin clusters, these green ones, assemble at the contact site. Cortical tension prevents this initial interaction site from spreading. The cadherin generate local signal to inhibit the GTPase Rho and activate the GTPase RAC, leading to localized disassembly of the actin myosin filament, loss of cortical tension, as you can see here. Okay, and formation of branched actin network and cell protrusions, all of which allow the recruitment of more cadherins and the spreading of the cell-cell junction over a greater surface area. In the long term, the large adherence junctions inhibit RAC and stimulates Rho, thereby promoting formation of local actin myosin fibers that interact with the cadherins. So now you can see when cell doesn't want to connect with the other cell and there is no such signal presented for the cell, then how the intermolecule or intracellular actin arrangement is like. When there's a little bit of signal for the connection with the adjacent cell, the remodeling of the actin leads to the formation of adherence junctions, right? This means that only having a surface representation of cell adhesion molecules is not the only requirement. The requirement is also for the remodeling of the actin filaments which are present in the cell. That is how cell is maintaining its shape and structure and allowing the connection with the adjacent cells. Now, if you try to connect this knowledge with the disease cancer and which leads to the metastasis of the cancerous cells, right? So for a cell to dislocate itself from one position to another position, we need to break these interactions. Then only a cell will be able to move from one place to another place, right? Here we are talking about cadherins, which are cell to cell adhesion molecules. So there are other type of connections which connect the cell to the extracellular matrix also, right? So in that case, this is how the cancer cell has evaded or is still prevalent, because they are able to relocate themselves by dissolving these connections in the present place wherever they are present, wherever the cancer cells are there, and get located to the other places. And again, if a cell is getting located or getting settled at one place, again they have to make these cell adhesion junctions and other type of connections with the new place wherever they are being transported. Okay, that is why understanding how cell-cell connection or cell-cell junctions is made is an important therapeutic target also. Okay. Now, catenins link classical cadherins to the actin cytoskeleton. What it means, so we'll see. The extracellular domains of cadherins mediate homophilic binding at adherence junctions. We just saw that, right? The intracellular domains of typical cadherins include all classical and some non-classical ones, interact with filaments of the cytoskeleton, actin at adherence junctions, and intermediate filament at desmosomes. These cytoskeletal linkages are essential for efficient cell-cell adhesion, and cadherin that lack their cytoplasmic domain cannot stably hold cells together. It makes sense, right? Because in the last class we saw that how cadherins interact. It's like a velcro like mechanism. So a cadherin on one cell will interact with the cadherin molecules on the other cell, get recognized, then it will lead to the intramolecular signaling, and that is how the connections is being made, established. The linkage of cadherins to the cytoskeleton depends on adapter proteins that assemble on the cytoplasmic tail of the cadherin. At adherence junction, the cadherin tail binds to such protein, beta-catenin, and a distant relative called P120 catenin. A third protein called alpha-catenin interacts with beta-catenin and recruits other proteins to provide a dynamic linkage to actin filaments. You guys don't need to remember all those things. The only information which you should take from this slide is that now we just saw that alone having only the cell adhesion molecules is not sufficient for the cells to stick together, right? We need these molecules to have a stable connection with the intermediate filaments, with the filaments which are present inside the cell, such as majorly actin. So this is what this particular slide is about, that the cytoplasmic domain of cadherins has the ability to get attached with different types of protein or act as adapter protein itself Few of these adapter proteins are alpha catenin and beta catenin. Third one is beta catenin, and different one is P120 catenin. So this will allow the cells to have a connection between the internal of a cell, that is the actin filament rearrangement, and outside of the cell, that is allowing the cadherin molecule of a cell to get connected with the cadherin molecule of another cell. Again, that cadherin molecule on the second cell is also connected with the actin filament of that second cell, right? So this is what basically it is like. So the linkage of classical cadherins to actin filament, this is the basic way how they are connected. So you can see that these are adapter proteins which are connected with the cytosolic domain of the cadherin molecule itself. So when the adapter proteins are connected to the cytosolic domain of this cadherin molecule, other molecules which will allow the actin filaments to get connected with the cadherins are also present. That is how a single cadherin molecule is linked with the actin of one cell, and imagine there is another cell here which has the similar arrangement and they are connected via cadherin molecules. That is how cell has the ability of contact. They know when there is a new cell or cell in their proximity and how much they should grow, how much space they should have. So these are the roles of the rearrangement of actin filaments itself. Okay, now we'll see a little bit of information about these cell adhesion molecules and how basically cells recognizes each other and how they behave when there is an adjacent cell. So just give me 1 minute, I'm gonna reshare it.
Video, I'll be talking about the adherence junctions. So in the epithelial cells, there could be multiple different type of cell junctions. There could be adherence junctions, there could be gap junctions, there could be tight junctions, and so on. So in this particular video, we'll focus on adherence junction and its physiological relevance and importance in development and cancer. So the adherence junction between two cells are just like something is hooked into a wall. So it's like, so there would be something anchoring region, and on the other hand side there should be some support, and we would be talking about that in this whole video. So here are the two cell junctions in a magnified version. So you have the cadherin molecules. In a second we will talk about cadherin molecules in details. And you have the actin microfilaments. So the actin microfilaments are the rigid supports, and the cadherin is one side attached to the actin microfilaments by several adapters, which are termed as catenins, and on the other hand side it is attached to another cadherin molecule. So there is a homophilic interaction between two cadherin molecules from two sides of the cell, and each of the cadherin molecules coming from the cells are connected to the actin cytoskeleton of the cells, right? Now, since one of the connection between these cadherin is very weak, there are multiple different connections between cadherins in array. So together, all of these connections make it a strong connection. Now, these cadherin proteins are connected to the actin cytoskeleton by catenins. One of the important catenin is alpha catenin, beta catenin, and also P190 catenin. So all these catenin molecules are working like an adapter, which is attaching the cadherin molecule to the actin cytoskeleton. But apart from its attachment function or adapter function, catenin molecules are important platform for signaling. For example, beta catenin is important for Wnt signaling. So this could have also signaling roles as well. Now, this cadherin has 5 extracellular domains, which are like in a jumbled situation when there is no calcium available, or when the calcium level is low. But when the calcium level is high, there is a conformation shift. So it becomes more rigid and less flexible, and they also dimerize to form a rigid structure, which can interact in cadherin molecules on the other hand side. And this homophilic interaction, though each homophilic interactions are weak, but together all the homophilic interactions make it a very strong junctions and connection between them. So cadherin basically connect the two nearby cells like a velcro. So each of the interaction between the velcro is weak, but together the velcro interaction is pretty strong. So this is a crystal structure of the cadherin, which shows the EC1 domain of the cadherin coming from the each side of the cells are interacting with each other, and the cadherin need calcium for their interaction. So it has been seen when the calcium is washed away or calcium is chelated, the adherence in vitro situation or even in vivo situation, the cadherins cannot interact with each other in a strong manner. So there could be multiple different type of adherence junction. Sometimes they are punctate, so they are known as punctum adherence. Sometimes they are like a zone or band of adherence junction known as zonula adherence, and there could be even complicated tricellular adherence junction as well. So we now talk about adherence junction, and it's important, and cadherin it's important in terms of the development. So we talk about the neural tube development. So we know after gastrulation there are three defined germ layers: the ectoderm, the mesoderm, the endoderm. The ectoderm gives rise to the nervous system, and the ectoderm is an epithelia, so the epithelial cells express E-cadherin. But the ectoderm just top of the notochord starts expressing N-cadherin, and otherwise known as neuroectoderm. So you can understand the neuroectoderm is just differentiated from the nearby ectoderm by the expression of specific different cadherin, N-cadherin, and the other ectoderm tissue express E-cadherin. So depending upon the expression of the adherence junction proteins, the tissue identity could be different. Now later on, when the neural tube is folding and neural crest cells are migrating out, they express different type of cadherin. For example, the region which is known as the floor plate, which is just above the notochord, starts expressing cadherin 6p, whereas the other cells of the neural tube express different cadherin, different N-cadherin. The migrating cells, which are neural crest cells, express cadherin 7. So depending upon the type of cadherin it uses, its adhesion properties are very different, and sometimes the adhesion remodeling can give rise to the cell migration and cellular change in cellular architecture. Now we talk about brain development. In the brain development, these adherence junction proteins, especially the cadherins, has a huge role. Now, when our brain is developing, it has something called ventricular zone. In the ventricular zone, there are mostly cells which are neuronal stem cells. They are progenitors. They would give rise to neuron and glia eventually. They are connected to each other by these adherence junction proteins, especially the cadherin 2. Now, what happens is these specific interaction between the cadherins are very strong. They allow these cells to divide continuously and increase in number before they can give rise to other particular neuron or glia. And these adherence junction based interaction also ensures that they don't leave the ventricular zone and mismigrate into other regions. So they hold still on the top of the ventricular zone. So what happens is this cadherin 2, which is one of the specific neuronal cadherin, is very important. In some cases where this cadherin 2 is mutated, there is drastic changes in the brain. So in the normal mouse brain, it has been seen there are apical progenitors marked here in green, and the basal progenitors, or marked here in the red, are separated by distinct zone. Now what happens in a cadherin 2 mutant mouse is the apical progenitor and the basal progenitors, their adherence properties are altered, so they kind of intermingle with each other and they move out to any location. That leads to a huge defect in layer formation in the brain, and the whole brain seems to be jumbled up. The whole cortex seems to be jumbled out, and eventually these animals die. Not only in brain development or neural tube development, in terms of cancer, if we think, then here is a epithelial carcinoma we are talking about, and then eventually the worst case scenario is the cancer cells migrating to other locations via bloodstream, and they can detach from the epithelia because their adherence properties are now different. It turns out that most of the cases, the expression level of E-cadherin in cancer cells are drastically low than the controls. That allows them to easily detach from the epithelial layer and move around the body and invade other organ, and thereby giving rise to a secondary cancer outcome. So this is how adherence junction and cell junctions plays a vital role in cellular morphogenesis from brain development, even cancer. So if you like this video, give it a quick thumbs up. Don't forget to like, share, and subscribe.
Yeah, so that was the overall view of how cadherins generally make the connection. Apart from this, as you know, that till now, I guess you guys have also started connecting the dots, that whenever we are talking about membranes and the proteins associated with the membrane, we are always saying that they are made up of weak interactions. If you look at the individual interaction, so it will be of weak in nature, but as they are more in number, the interactions are more stable, and hence the system is stabilized with lowest energy possible. This is the beauty of the biological system, right? So individual interaction is weak, but if we sum up all the interactions, all the weak interactions together, they are very stable and they are very strong. So why we should have such weak interactions in the first place? Because our life is dynamic in nature, right? We can't have all the cells of same size. We need different cells of different shape and size. So if we put a strict geometry on the cells, that they can be only of spherical in nature or they can be only of elongated in nature, then most of the functions which the cells are adapting themselves to perform, they won't be able to perform. So the benefit of having these weak interactions in multiple numbers is that whenever there is a need for remodeling of the cell or the adaptation of a cell depending upon external or internal stimulus, it becomes easier, because you need to rip out the weak interactions, though multiple in numbers, but they are weak in number, and hence the cell doesn't need to put that much of energy in breaking those stable connections in comparison to when if cell makes a very stable connection with a strong bond such as covalent bond. Okay, now see, these are just how we are discussing about the adherence junctions. There are other connections also between the cells, right? As we have seen in the beginning of last class, that there are n number of connections between cell to cell, cell to the surface, to the basal layer. So all those things perform different functions. So right now, where are we? We are basically discussing about the cell-cell adherence junctions. So this is another function of adherence junctions itself. So adherence junctions respond to tension from inside and outside the cell. This is pretty understood in itself, right? If cadherins molecule which are making the adherence junctions, they are connected via adapter protein, and those adapter proteins are connected to the actin filament, which clearly indicates that if there is any sort of tension or mechanic stress inside the cells, the cell surface molecules such as cadherins in this case will be able to sense it, right? So we'll discuss this property in the next class. Till then, try to read about cell adherence junction, and you will be fascinated how membrane proteins not only allowing the transportation, but they are also involved in connecting one cell to another cell, and even in the connection they are allowing some type of transportation and connection between the adjacent cells. Okay, so thank you so much for your attention, and we'll meet in the next class that will be on Tuesday. Thank you.